Well, good afternoon and welcome to this Dean's Research Seminar. I'm John Pasackley, I'm the Dean of the Faculty of Veterinary and Agricultural Sciences at the University of Melbourne. And I welcome you to this seminar, which will be given by Professor Mohan Singh. But before we begin, I'd like to acknowledge the traditional owners of the lands I'm on today, the Wurundjeri and Boonwurrung peoples, as well as the traditional owners of the land you are situated on, and acknowledge that Indigenous Australians have been custodians of the lands and waterways of Australia for tens of thousands of years, and I pay my respects to their elders, past and present. Today, Professor Mohan Singh from the School of Agriculture and Food will present the Dean's Research Seminar with the title Synthetic Biology for Biodesign of Crops for the Future. Professor Singh, Mohan Singh, completed his PhD at Punjab Agricultural University in India. He's had a long career at the University of Melbourne from his arrival in 1982 on a research fellowship to becoming a full professor in 2005. Since then, he's held many leadership positions in the faculty, including periods as Deputy Director of the ARC Centre of Excellence in Integrative Legume Research, the Associate Dean for Research, the Director of Food Systems Programs, the Acting Head of School, the Deputy Head of School and Director of Teaching and Learning. He's also contributed to over 40 university, faculty and school committees. The focus of his recent research is to use synthetic biology and biodesign strategies to develop crop genotypes that can sustain successful reproductive growth and seed yield under heat stress conditions. Mohan was one of the pioneers in the application of molecular biology tools to understanding fundamental aspects of plant sexual reproduction. He has an H index of 52 with more than 190 papers in international refereed journals and 50 published book chapters. His research on biotechnology and its implications for agriculture and human health has been presented at conferences and covered in print and broadcast media. He's the inventor of 58 granted patent applications in allergy and diagnostics and therapeutics, genetic engineering of male sterility for hybrid seed production, and novel male gamete-specific promoters. He's trained 30 postgraduate students and 40 postdocs, and many of these have gone on to become successful leaders in their fields. Amongst his measures of esteem, Mohan has been awarded a Thyro Medal for his research achievements and the Frederick White Prize of the Australian Academy of Science. Please welcome Professor Mohan Singh. Mohan, over to you.
Thanks, John. Let me share my screen. Check. All good now?
Yep, all good.
Thank you. Thanks, John, for your kind introduction. And I love your canola background in your Zoom, because this is something which is going to feature a lot in my presentation today. But let me start my presentation by showing you what Nobel laureate Paul Crutzen has said. And he said, this is the era, geological era, Anthropocene, which is a biosphere shaped by humanity. And let me talk about, with highlighting global challenges to sustainability of health of our planet, as well as, feeding that growing population in a stressed environment. So basically, we as humans are changing the face of the earth. And there are multiple challenges posed by increasing urbanization, as you can see, water shortages, fires, deforestation, and so forth. But there is no bigger challenge for us than feeding the growing humanities. And this challenge is even made bigger because as population is growing, people are becoming more affluent. They are not looking for just more calories. They are looking for high quality foods, more protein, which puts a pressure on us to produce more feed for the animals. Okay. And then also consider challenges posed to food supply chain by geological conflicts, wars. As you know, current war in Ukraine is causing havoc with the food security, right? And food price inflation is reducing people's capacity to basically acquire food. So we in the SAF, have a mission to work on those grand challenges. And I was pleased to note that my last 3 speakers in the same series, have been focusing on such big pictures. And let's focus on big pictures as far as plant biologists are concerned, what we can do to enhance food production, sustain food production in face of changing climate. Now, climate change is real. Climate change is affecting food production globally. And you can see that global south is facing more challenge. And another point is uncertainty. So year-to-year climate change is making crop production uncertain. And this map shows kind of prediction for 2003 to 2080. And it has been estimated that there will be 20 to 49% yield fluctuations. And it can happen year to year. And this is, a serious issue. So what can plant biologists do in the face of these scenarios? Now, this has been the news for last 3 to 4 months. And almost probably all of us have read somewhere or the other that the heat wave in India has affected the wheat production by more than 10 to 15%. India is actually wheat exporter, and they were hoping to export a lot of wheat, considering also there is a food shortage caused by Ukraine war, and they were hoping to earn a lot of foreign exchange, sell the wheat. But just when crop was ripening, the crops were in a stage when you can say early seed development, reproductive stage, here comes heat wave and basically scorches, as they say, wheat crop, loss in wheat productivity. And also there was a news, also I can see very recent, that there was also in Canada the same thing, because Canada is relatively colder country, we don't expect that, but heat waves are being experienced there as well. We in Australia are not, away from this, because here is the map for last more than 100 years, the warming. So our climate is also warming. Look at 1919 and 2019. And 2018 and 19 has been the warmest years for Australian climate. And here we can see, and there is one more issue, frequency of the extreme heat events is increasing. So, and this can cause serious issues with food production, and particularly if these heat waves happen at the time when plants are reproducing, seed set, pollen development, all those plant developmental stages are highly vulnerable. So in my presentation today, I'm going to focus on 2 issues. One is, can we develop stress tolerant crops? And I will be basically talking about heat stress today. And I'll also segue on to pollen allergy, because allergies are also increasing with the climate change. And this has been our long-term interest, as John said, pollen allergies, vaccines. So I will first start with stress tolerant crops. So this is our lab. The actions happen here, and we have a website. So please, everybody's welcome to look at our research programs. And our research focus is on a lot of crops. And today I'm going to focus on canola and a bit on soybean and grasses, because grass is more in the context of grass pollen allergies. Now, just to remind you that all the grains we eat, food we eat, plant foods, are product of sexual reproduction. Plant has to flower, produce pollen. Pollen has to fertilize the egg, start the seed set, and that's what we eat. So in case of rice and corn, it is the endosperm of those grains is our food. But in dicot crops like soybean, it is cotyledons. But nevertheless, it is the storage material in the seeds and grains which becomes our food source as well as source for animals. Now, sexual reproduction is a stage which happens in the very late stage in the pollen in the plant growth. First, plant has to grow vegetatively, and then it starts transition to flowering. Flowering means further development of male and female reproductive organs, male reproductive organ, which means, and then female reproductive organ. We think about here stigma, style, where the fertilization takes place and pollen tubes go down and fertilize ovules, and also here reproductive synchrony is very important. Here we see a normal canola flower reproductive organs, female organs, and anthers, which are male organs. And here, after a heat stress event, the anthers haven't grown much. First of all, sterile pollen and incomplete growth of anthers. So this puts all the spanner in the works for reproduction. And if there is no sexual reproduction, no food. So this is important. But these stages are highly vulnerable to abiotic stresses, more viable to heat stress than any other stress. And that's what happened in Indian conditions, that a lot of productivity was lost during the reproductive stage in case of wheat. So we have been focusing on canola as using our model system. And here we've done experimental work, which has been published in Journal of Agronomy and Crop Science. Here we show that exposure for few hours at varying temperatures, 30 degree to 40, has serious effect on pollen viability. Canola normally grows at 25 degrees C or so, but anything more than 27 degree really reduces the productivity. And here we can see that in 4 hours at high temperatures, 38 or even much higher temperatures, there's a dramatic drop in pollen viability. More than that, germinability, whether pollen can germinate and actually penetrate the stigma surface for fertilization and seed set, is even more vulnerable. We end up with a lot of empty, shriveled calyx. Calyx are the fruits in the canola. We call them calyx. So here we can see number of empty calyx percentage increasing at even if for 10-hour heat at 40 degree, basically 30 to 40% calyx are totally empty. And also serious decline in the seed yield with increasing ambient temperatures. So we are talking about short-term heat stress, even for one day, ranging from 4 hours to 10 hours, and then can have serious implications on canola productivity. And we are simply showing here how pollen looks like. After 4 hours of heat stress, about 60% of pollen have become non-viable. And if you say 10 hours of heat treatment for 1 day, complete loss of viability. This is viable pollen developing in the anthers, which are the male reproductive organs where pollen grains are formed. And after a heat shock, basically most of the pollen have become sterile. And you can see here pollen viability as tested by fluorescence, we call it fluorescent diacetate test, and loss of viability, and you end up with very little, pollination, fertilization, and hardly any seed set. It's an extreme case scenario, but it can happen, right? Now we're talking about reproductive synchrony of development. So this is a canola, dissected out canola bud in the bottom, and the control plants, the buds look like this, very healthy anthers. And moment, even if 1 day extreme heat event, we examine 34, 36, 38, 40, and what you see here is effect on growth of the male reproductive organs. Not only loss of pollen viability as such, but here we are at 40, even the anthers are shriveled, pollen will be, yeah. So you can see here, and also loss of, seed production is so different here, and basically shriveled seeds, and so 34, 40. And also, we have also examined oil quality of these canolas, which are under stressed. Interestingly, the omega-3, omega-6 ratio changes. So omega-6 goes up by, say, about 10%, and omega-3 level goes down by similar amount. So there is effect on not only on seed development, oil production, but also oil quality. So what we want to do, we want to look at whether we can engineer canola, which is more tolerant to heat. For that, we have to understand molecular basis of pollen development. What gene expression programs are linked with pollen development? And before we can do something with the heat, let's look at the normal pollen development. And on the left is BCP1 gene, and I'm highlighting there because this was the first gene discovered which controls male fertility. The gene is expressed in the anther wall as well as pollen, and when we knocked it out with antisense technology, that was the technology available nearly 2 decades back, and here we get complete sterility in a model plant Arabidopsis. Now, recently, last 2 to 3 years, we have shifted to looking at long non-coding RNAs or non-coding RNAs. And we have now discovered first long non-coding RNA, which is essential for pollen development. This long non-coding RNA doesn't produce any protein, but somehow it regulates pollen development. Here we are. If you can see here, pollen tubes growing in the stigma tissues. This is control pollen with the control stigma. And here, if we have a normal, this mutant, so we produced a mutant Arabidopsis plants with CRISPR-Cas technology to knock out the gene which codes for this particular long non-coding RNA. and we get sterility. So that shows that it's not only the protein coding genes, but non-protein coding part of the genome is equally essential for reproductive development. And let me go into more detail. Many years back, it was the genome which was coding for protein was considered the functional genome. And I have read papers nearly 2 decades back when they call this part of the genome as a junk DNA or junk genome. No longer. We know now that it is the dark matter of the genome transcribes a lot of different kind of RNAs, which play important role in development, reproduction, growth, and long non-coding RNAs, we classify them as transcripts which are more than 200 base pairs long and do not have any coding potential. But they are emerging as, growing players in development, also in abiotic stress tolerance. Just to give you some example how the long non-coding RNAs can affect development by they may not be producing protein themselves, but they can affect other protein coding genes. If long non-coding RNA locus is next adjoining the protein coding gene, this is called cis interaction. So it can either suppress the gene expression or even enhance the gene expression, depends on the context. Or long non-coding RNAs can work as sponges for microRNAs, and which instead of microRNA affecting the mRNA, it's going to be sponged away by long non-coding RNAs, or even decoys for transcription factors. So, or even scaffold for different proteins. So long non-coding RNAs are coming as, very important actors in controlling plant response to abiotic stress, plant development, as we have shown example in our own lab, the Arabidopsis, where we have shown that one of the long non-coding RNA expressed in Arabidopsis pollen is essential for male fertility. And by the way, that particular RNA, long non-coding RNA we discovered, is trans-acting RNA, but I wouldn't go into details. I'll show you something more. Circular RNAs. So last about 3 years, one of our PhD students has been focusing on circular RNAs. Circular RNAs are basically RNAs which make backfolds, and they can come from introns, exons of the gene, different, or from non-coding, 3' end. And they have to be, when you sequence the circular RNAs, you have to verify them by sequencing that the splicing has indeed happened. And there are some examples from our lab. Circular RNAs also seems to have stage-specific gene expression in pollen. And this is during normal development. And now we'll be also focusing on development during heat stress. But most interestingly, we have been studying floral transition in soybean. And we have sampled the short apical meristem at the vegetative stage, and later on floral inducted stage, made libraries for circular RNAs, sequenced them, and we have discovered now few circular RNAs which seems to play a very essential role in floral transition from vegetative to reproductive stage. But so talking about long non-coding RNAs, we started this work about 5 years back, and we have been highlighting the role in abiotic stresses response to pathogens, but more importantly, in reproductive development. And one of the opinion we published, we have shown that long non-coding RNAs are key players in reproduction in plants and animals. In plants, they are highly expressed in pollen, anthers, plant sperm cells, and in also animals in testes and other male reproductive organs. So there is so much evolutionary convergence of the role of these long non-coding RNAs. And we have also identified long non-coding RNAs, which are specifically expressed in the sperms in the rice pollen grains. Long non-coding RNAs have been shown to have implications with the agronomic trait. And here we did genome-wide analysis, and we have identified long non-coding RNAs, which are possibly linked with agronomic traits, or we can call it yield traits, such as days to flowering and from flowering to maturity and number of seeds per pod. So long non-coding RNAs are also, seems to play very important role in reproduction. In case of, our focus is on pollen, because pollen is the most sensitive tissue or cell to heat stress. We get, as I shown you pictures, we get male sterility with even one heat event, even 4 hours, even 6 hours, and we get sterility, and that has serious effect on crop productivity. And here we have got different stages of pollen development, but this is at the normal temperatures. We can still see that as compared to coding genes, a lot more long non-coding RNAs are expressed. And we do not know the role of all of these, but because you had to basically, as we did, knock down the genes and look at what happens. But we can clearly see that even more than coding genes, long non-coding RNAs seems to play a clear role in stage-specific development. One thing I like to add here, that as compared with the protein coding genes, long non-coding RNA transcripts are not highly conserved. So if we find a long non-coding RNA in brassica pollen, it might not be conserved in other crops. So they're less conserved, but they have very, very important roles in development. Now, we have been talking about problem of heat stress caused pollen sterility and how to address it. Is it possible for us to genetically engineer crops in such a way that they can withstand more abiotic stresses? So this is a result from our PhD student who worked in a ASRF collaborative project with International Center for Genetic Engineering and Biotechnology. So here we overexpressed gene encoding DNA helicase. Now, DNA helicase is, as shown in a very simple model, is responsible for opening up the helix. So breaking the hydrogen bonds with the use of ATP. And this opening up is very important for both DNA replication as well as transcription. Interestingly, if we overexpress the gene taken from a pea and put it in the canola, we see very good salt salinity tolerance. So here are the canola plants growing in 200 millimolar sodium chloride. And here we can see control plants shriveling, chlorosis, loss of chlorophyll, and the plants which are genetically engineered to overexpress P. helicase canola plants are doing relatively much better. And this simply shows a protein dot plot showing that high helicase expression in transgenic lines. Interestingly, when we tested the same plants, PDH 45 overexpressing plants in the heat stress, and we gave 1 week heat stress with gradually changing temperature every day and going maximum up to 35 C. And we saw that the plants which are overexpressing helicase has much better protection from heat. And you can see here the pods with some seeds as compared to hardly any seeds in case of control plants. So there is a protection with overexpression of PDH helicase came from P. And you can clearly see here, transgenic plants versus control plants. So there is a clear correlation. Now, thing with helicase is much more broad, works broadly. We need to look at genes which are much more focused in the tissues which we are looking at. And this is just a general model of heat sensing networks. We have big players before, and also what heat does, it produces misfolded proteins in the cell, and also endoplasmic reticulum stress, which is known as a unfolded protein response. All these responses lead to heat sensitive gene expression. Let me spend some time on PIF4. You have seen that in a previous presentation that overexpression of this particular gene changes reproductive transitions in soybean. But more interestingly, we have recently studied heat tolerance in the transgenic plants. So here, I just want to show you here soybean flower. Soybean flower has a very distinct shape. The male stamens are fused as a staminal tube, and this is the control. And under heat stress, the anther staminal tube becomes smart. Here, sterile pollen anthers and a smaller tube. But the PIF4 overexpressing plants are showing much better. They are not as equal to control, but they're still much better protection from heat. And also, overexpressing plants showed higher seed yield under heat stress conditions. So now let's go look at the problem again with PIF4 is PIF4 is a transcription factor which binds to activates number of different proteins. If we want to develop a heat stress tolerant pollen, we have to look at single gene which is expressed only in the pollen, and we can upregulate. And recently we studied this part of the pathway by giving heat stress to pollen and looking at the transcriptome. Most interesting observation is that 40-degree heat stress for just 5 minutes. We just give 5-minute treatment and then study transcriptome or 15 minutes. 5 minutes heat stress was more than enough to completely change the transcriptional profile of canola anthers. And here we can see the genes which are in the green switching on in 5 minutes. We call them early responders. And so all it shows is that there are possible candidate genes which are early responders. We may be able to overexpress them to get what we want. Heat stable pollen grains. That's the ultimate goal. So as I'm talking about heat stress susceptibility, canola is heat stress from flower opening to week after flower opening, week before flower opening. pollen development stage, fertilization, all those things are. This is the field condition when heat stressed plants, basically empty. And this is again in the field stage, the healthy, normal canola, heat stressed canola. So we have identified doing transcriptomics possible candidate genes which can be overexpressed to get the desired effect. But the challenge is that protective genes have to be expressed at the right time in the right tissue and only during heat stress conditions. And that's a challenge because many of the proteins are genes are heat inducible. Proteins, long non-coding RNAs, they're heat inducible, but they may not be only induced in pollen or anthers, probably in a lot more tissues. And we need genes which are anther specific. So one possibility for looking for, so basically we need a promoter element, a controlling element, which expresses the gene in anthers, but only under heat stress conditions. So what we are doing now to look for that promoter, to look at that regulatory element, we are searching our transcriptomic data if there is any native gene with such an expression pattern, which is pollen specific and heat inducible. That is the one way of doing it. But these days, the future now in biotechnology is synthetic biology. People have been successful in a lot of other situations, and we are looking at developing synthetic promoters, taking cis elements from different promoters, combining them in a combinatorial way to develop that kind of promoters which are heat inducible in anthers only. So that's the ongoing work. So I will segue into something totally different here, which I talked about, allergies. Allergies are particularly pollen allergies. And we have been working on grass pollen allergy for more than 3 decades. And I'll explain to you what we very briefly, what we have achieved. But most interestingly, with climate change, I have just a paper here, which is shown a paper here published in PNAS, which shows anthropogenic, climate change is increasing pollen levels in the environment. Because of the weather patterns, more heat, plants, grasses are producing more pollen than ever before. And grasses basically release pollen in the air because they are wind pollinated. And these basically, when we talk about allergy is a hypersensitive reaction. And when our immune system reacts to allergen by producing immunoglobulin E antibodies. Also, rainfall events in the pollen season enhance the risk of something called thunderstorm asthma. And we'll understand thunderstorm as asthma in following slides in my presentation. But firstly, I'd like to share with you what are grass pollen allergens. What are these proteins? These 2 proteins have been cloned in our lab. One of them are published in PNAS and other journals. But more importantly, we have got intellectual property rights and patents for them for use of these proteins for immunodiagnostics and also for developing vaccines. So pollen grains, when they get into our respiratory system in the hay fever season, when the grasses are flowering, they release proteins, number of proteins. Out of them, 2 of them are major allergenic, which means they elicit formation of IgE antibodies and downstream allergic reactions. One of them is a LOLP1 simply called because of the name of the plant, Lolium perennial ryegrass, LOLP1, it's a 35 kilodalton, and other is a smaller protein. The fact to remember here is these proteins are highly conserved between different grasses. So if you're allergic to ryegrass, you automatically become allergic to a lot of the grasses. So that's why we call it grass pollen allergy. We don't call it ryegrass pollen allergy or Kentucky blast pollen allergy, because they can be 80 to 90% conserved, and their IgE binding epitopes, the regions, are highly conserved. And I want to show you something very more interesting, that something about asthma. Or when we clone these two genes, we if the pollen falls on the female stigma surface, here the pollen tubes growing, that's the normal function. But we cloned two genes to identify two proteins, and we did immunolabeling experiments where we labeled the monoclonal antibodies to different size of gold particles. And this shows that one of the protein, LOLP5, is present in the cytosol, whereas LOLP1, one of other protein, is present in the starch granules present inside the pollen. So before we did this work, there was a great conjecture in the literature how pollen can cause asthmatic attack, because normally the particles had to be less than 5 microns to go down deeper in the lungs. But pollen was shown to cause allergic reaction. And this was a very iconic, basically, photograph for us. One day, and the basically ryegrass came from backyard of our home, and we tried to do this kind of scanning electron microscopy. And one of our technician, when he forgot to put osmoticum in the fixative, and he came to us saying, this is what I'm getting. And we said, they are particles. Maybe they are those micronic particles. Maybe these are the one, because if pollen comes in contact with water, if it doesn't go in the stigma surface, it's basically coming in water with not proper osmoticum, rainwater might explode just like that and produce 500 to 600 small particles full of allergens, and they can go in the lungs. And actually, later on, a French author have actually studied that particles, they are between 0.6 to 1.6 micron, perfect size to penetrate the stigma surface. And in fact, and this was published in Lancet, and in fact, this is now accepted fact, and Department of Health has produced a video. Some problem here. I will not be able to, it's not going on, but there is a video which shows how pollen grains burst in the rainy season. When the pollen grains in the air, they burst and release the micronic particles. And here you go, particularly during thunderstorms, during the allergy season, there are a lot of hospital presentations, so particularly people who are susceptible to asthma. And it has been in the news even last allergy season in Melbourne. So the knowledge which we came from, just serendipitous experiment thinking, it has been now accepted all over the world, and people have experimentally verified this is where the particles are coming from. Apologies for not activating the video here. Oh, it is working. Sorry.
In Southeast Australia. These events can be sudden, serious, and even life-threatening, and many people may need help at the same time. So how does this happen? Pollen grains from grasses get swept up in the wind and carried for long distances. Some burst open and release tiny particles that are concentrated in the wind just before the storm. They're small enough to go deep into the lungs and can make it difficult to breathe. This can become very severe very.
Okay, we can't watch whole, not enough time for us to hold whole video. But our question is, if we have identified those proteins, why can't we make a vaccine? And this is interesting. This is very original paper in Lancet, 1911. Dr. Leonard Noon from St. Mary's Hospital, London. He experimented on himself and some people by injecting pollen extract, because at that time, there was no idea they are the proteins. They call it pollen toxin. And he made a aqueous extract and injected himself and some other people. And he found that if you do that before the hay fever season, the sensitivity to pollen goes down. Call it prophylactic inoculation against hay fever. But can we make, but this is a very crude way, and it is still being used in a way. The pollen, extract injections, they're more standardized, but this is not a designed vaccine. Can we design a specific vaccine? Because if we inject this extract in people, we are possibly enhancing the chance of, becoming allergenic as well, right? So we need to develop. So one idea was to map, now I won't go into immunology, map B cell and T cell epitopes of allergens and modify B cell epitopes so that they can no longer bind IgE, but bind other kind of antibodies. Immunoglobulin 4, for example, that's a blocking antibody. That was the principle behind our research for making specific allergen-specific immunotherapy. And some of the candidates we designed, we just created few mutations based on our study with using synthetic peptides. Here we have mutated proteins with, and also showing that T cell activity is maintained and mutant pollen have, reduced sensitivity in terms of skin prick test. And this test was done in Vienna by our colleagues. And here we can show a candidate vaccines. Also, we made mutants of another protein, LOLP1, previous LOLP5. And if we replace cysteine residues with serine in terms of mutation, at least in this part of the protein, we can see serious reduction in IgE reactivity, which means allergic potential, which is moving towards possible candidate vaccines. So I want to show you that our lab has contributed. In fact, I found a report which is produced by Australian government, IP Australia, the patent analytics report. So our lab has contributed more than 100 patents on pollen allergens. And they all have been funded by Merck or Allergo Pharma. So I stop my presentation here by acknowledging a number of students and postdocs and other collaborators, particularly listed their individual contribution. A lot of talk about canola heat was done by work was done by Dr. Nita Lohani, who just finished PhD recently. Dr. Hina Arya has been working on soybean PIF function and fantastic data on heat stress tolerance. Syed is a PhD student working on circular RNA and others so forth. I have listed there and also past co-workers who are not listed here. And Professor Rudolf Valenta was our key collaborator for doing skin prick test and all those things, and also project joint with ICGB, and also advice from Professor John Maddick from UNSW UQ and Professor Zuji Wank. And Allergo Pharma and Merck were basically funders of all those patents. So I finished my presentation here. Thank you very much for everyone for listening. And I'm open for questions. Thank you.
Thank you, Mohan, for that really interesting talk. I learned a lot. Perhaps you could stop sharing and we will open this for questions. Would people like to put questions in the Q&A, and you can ask them personally if you want, or I can read them out. Let me start, Mohan, a couple of questions that occurred to me while you were talking there. And the first one is it particularly grass pollen that explodes in that way and produces these small bundles of proteinaceous material, I suppose, mostly that goes down into the lungs, or does all pollen do that? And
looks like that pollen of birch, which is an important allergen in Europe, has been shown to be like that. It is simply pollen are full of osmotic, activity material. It is just simply osmotic busting, because unless they get onto the stigma surface, there is no such quick hydration. Basically, once they hydrate, they are so full of material with the pressure, they burst. So that's what happens.
So pretty much all pollen bursts open in that way.
At least they have shown experimentally in birch. I'm not sure.
In birch trees, okay.
Birch trees,
Yeah, okay. I was just wondering why grasses in particular, like, because the people that are allergic, I mean, it is called hay fever, which is another way of suggesting that it's grasses that are particularly important for the reaction that people have, either be it the asthma or be it a nasal reaction. But I guess people are also reactive to pollen in other plants. But I suppose it's mostly grass, and that's because they burst open like you've described, I'm guessing. Okay. My other question I had was, you said you were searching for temperature-specific promoters that might be active, particularly in the anthers.
Yes.
And you didn't really go into this, but in animal cells, and I guess in bacteria, you get these heat shock proteins, and I guess you do in plants as well.
Yes.
Because one of the main functions of proteins need to be folded up correctly, and when it gets too hot, they don't fold correctly, and so you have to have these heat shock proteins to make them fold properly. But can't you just use a heat shock specific promoter, and wouldn't that work for much of what you want to show?
Yes, the fact is the heat shock promoter is probably active in a lot more tissues, and you don't want to do that. So, but we are searching for. We just done a lot of transcriptional, study looking for the heat shock factors or heat shock proteins, or essentially any other gene which is expressed in pollen in response to heat. Then we can take that promoter part and express any gene. So I think this is an ongoing work, but we are confident of working here.
Yeah, and are you? I don't know anything about this, but I was really interested to hear what you had to say about it, and that's the long non-coding RNAs and the circular RNAs. Is the nature of their promoters very different to the nature of a promoter for an RNA that actually codes for protein?
No, nature of protein promoter is not different. In fact, I went a bit quickly. The Arabidopsis long non-coding RNA, we've taken the promoter and fused with the reporter gene, and we can see specific expression. However, the reporters are less stronger because they are expressed at lower level.
But they're long non-coding RNA promoters.
Yes, but they are expressed at lower level. However, they're highly tissue specific. So again, that's another source of possibly cis elements for us.
Yeah. And you could perhaps change that synthetically to make it a stronger promoter.
Exactly.
Yeah. Okay. Well, really interesting things. Now I've got one question in the Q&A. Comes from Nicole Reichert, who says, thank you for the presentation. Unfortunately, she has to leave, but she's interested to know if you can control and reduce heat stress in canola through genetic manipulation, and whether this, if you can change the heat stress in the canola, does this impact positively or negatively on the nutritional profile of the canola?
Exactly. I think depends on, that is why we need a very specific promoters and very specific tissue specific protection. We don't want foreign genes or anything to expressed in, for example, seeds, where it can affect a nutritional quality. So we don't think so there will be any effect on nutritional quality. We should be able to, because we have shown that in fact it is possible to reduce say heat sensitivity in PDH experiment. Is simply we're looking for more precise tools. That's all we're looking for.
Okay. So those of you that are specialists in this area might like to come in and ask Mohan a question. But I had another thing that was puzzling me, Mohan, which I'll just move on to. And that is, you said that if you used a DNA helicase, that in, I think it was soybean, but also in canola, if you express the DNA helicase,
Yes.
that would actually give the plant some protection against salinity stress and also heat stress. How does that work then? What is that
Yes,
DNA helicase
that's a very thank you for interesting question. It looks like we have not done that because we have collaborators have done transcriptional profile of the plants overexpressing PDH, and they find that a lot of genes overexpressed are those which relieve oxidative stress. And oxidative stress is a common feature of all the stresses when accumulation of free radicals, high oxygen free radicals. So enzymes like peroxidase, superoxide dismutase, they seems to go up, and looks like the protection is coming. That is why we are getting salinity tolerance as well as heat tolerance, because it has to be something which is common between different stresses, and it looks like it is relief from oxidative stress. That's what it appears, but it's a hard, but only says there is a helicase has been shown. This is not only first, this paper was published in PNAS. A lot of people have repeated in different crops, and they are consistently showing the same result. And all they can do is do transcriptome and show what goes up, because helicase is you can't say big tool. It's not a very specific tool. So that's why we are unlikely to get the permission to grow, these through regulatory bodies. We need a very specific tool. That's why otherwise helicase is doing the job.
I mean, these are long and complex experiments, and I
Exactly.
understand that not very many people can actually, or it was quite novel when you started genetically modifying crop plants as opposed to Arabidopsis. But
Yes.
could you overexpress superoxide dismutase in these plants, and does that give them any protection against these abiotic stresses?
That's a very good question. When we did our recent transcriptomic analysis of 5 minutes heat stress, even in the anther, we are seeing ascorbate peroxidase, for example, which is same gene in terms of reducing the oxidative stress going up. So basically what pollen is doing, trying to protect itself by enhancing the expression of certain protective genes. So this is how we pick up protective genes. We pick up some of those and overexpress them more. So exactly, you are right. So one of our goal is to overexpress one of these oxidative stress protective genes in the pollen and active in the heat stress only. Yes, it is possible.
Okay. All right. Actually, we are out of time, Mohan, and I don't see any other questions in the Q&A. So it just remains for me to thank you for that really interesting talk. As I say, I learned a lot, and I'd like to ask you more about circular RNAs at some point.
Thank you.
But perhaps not just now. So thanks so much, Mohan. And to the audience, I'd like to thank everybody for taking the time to attend today's presentation. And I hope to see you again on the 9th of August for next month's Dean's Research Seminar, where Professor Michael Taus, the new director of the Victoria Drought Hub, will present his research on adapting to climate change and drought. Are stress-tolerant plants the right goal? So very much on a related theme. So I hope to see all of you then. Thanks very much.
Thank you.